So in general, what happens with bacteria in the soil is that they will extract, free nitrogen from the air, will fall to the soil, and that'll make its way to the bacteria. And the bacteria will start to fix that nitrogen, where they are essentially converting free nitrogen from into ammonia. And they do this with the addition of hydrogens to the original nitrogen molecule. Another structure that's formed is called an ammonia ion. So either way, ammonia is being produced as well as ammonia ions, and those are being released into the soil, which can change the pH of the soil, making it more conducive for plant life to grow and thrive. And then you have animals that come and eat the plants, and then those animals secrete also ammonia products into the soil, and that also perpetuates the kind of nitrogen cycle, because each different organism are relying on relying on nitrogen being fixed in order for different elements to be used in terms of developmental growth. So kind of an interesting process. Let's look at a diagram of it. So remember on the last presentation I showed you an image of the nitrogen cycle. Well, here you're seeing it in kind of a multitude of forms. So we have nitrogen in the atmosphere that's able to make its way down into the soil, and as it goes into the soil, it meets the bacteria meet it, and then the bacteria will fix that nitrogen. Now, the bacteria are located in the soil, as well as they can be found in the roots of different plants. So this is a legume plant, and the legumes have these different nodules on the outside of them, and that's where the bacteria can reside. And so once these bacteria come into contact with the nitrogen, they convert that nitrogen into ammonia, which can be used by the plant. So the bacteria in the soil will also undergo the conversion of the nitrogen from the atmosphere into an ammonia product, and that helps with the production of nitrites, which are produced by what we call nitrifying bacteria. And all that means is that this bacteria will it fixes the nitrogen by converting it to ammonia NH4+, but then it goes through the process of nitrification, where you have the production of nitrites. So that's all they mean by nitrifying bacteria. Essentially, these guys are the same. You just have nitrogen fixing, you produce ammonia. When you nitrify something, you produce nitrites. And then the nitrifying bacteria will start to produce nitrates, and then these nitrates can be used in the process of denitrifying bacteria, or in the process of assimilation for the plants to use. And then the plants, after they use these particular nitrates, then they can also release this out into the soil, or they can be broken down by saprophytes such as fungi, which also are going to produce more of the ammonia to continue production or perpetuation of this process. This animal right here is also going to release ammonia products, which can assist in the breakdown and ammonification of these elements into the soil. So our soil is essentially fertilized and made fit for growth of plants, or for the continuation of life with bacteria residing in it, through this ammonia production that takes place because we use free nitrogens from the atmosphere to enable this process. Now, the denitrifying bacteria, they typically produce our free-floating nitrogen that exists in the atmosphere, and we need that, obviously, because it perpetuates, again, this entire cycle. And that, again, is our image of our root systems that have these nodules on them that can harbor that nitrogen-fixing bacteria. So let's talk about some other soil microorganisms that kind of play a role in assisting in the fertilization of soil, which can be used to enhance life. We've got bacteria, fungi, algae, protists, viruses, viroids. All of them decompose numerous types of tissue. They assist in the breakdown of any living organisms that eventually die. And they help to return those organisms that do die back to their original forms, which are chemical elements. So in doing so, they assist in, the breakdown of waste products in some form or fashion. There's also a number of organisms that reside in the soil that enable us to study them because we can extract them from the soil. But things that can induce infection or be pathogenic to us have origins tied to the soil. Some examples are some of the Clostridium species, such as Clostridium botulinum and Clostridium tetani. Many of these are very dangerous to humans, especially if they've been consumed, and the reason for this is because they express certain exotoxins and endotoxins that can be lethal when ingested by a patient. The same thing with Bacillus anthracis. Bacillus anthracis is a rather scary organism just because it forms spores, which are very hard to kill, very hard to eradicate. And then these can be aerosolized, which means you can breathe them in, and then you can have a very lethal inoculation just from the Bacillus anthracis spores. So those things can be very, very scary for us, but we know that they reside in the soil, and because they are in the soil, we are able to go in the soil and cultivate some of these organisms, so hopefully we can better understand them. Okay, so lastly we're going to talk about biotechnology, since that's what this unit was also included. With biotechnology, I think I talked about this also in the first lecture that we did. You essentially have the use of technology mixed in with the use of biology, or the field of biology. So we use biological systems, living systems, or any combination of those systems to produce items that can enhance our own lives or make the world a little bit easier to live in. So it can be in the form of a product that gets produced, or it can be in the form of a process that might be followed. I think one of the examples I gave in the last lecture was the idea of using cells and a 3D printer to construct an organ that can be given or transplanted to somebody in need. So things like that is what we mean when we're talking about biotechnology. And specifically microbiology, biotechnology, they use bacteria, fungus, protists all the time, algae. They'll use algae to try to produce biofuels, so that way we can deplete our dependency on oil in the world, or pretty much all over, because the U.S. is not the only country that taps into this type of research. We use microbes to produce vaccinations, produce antibiotics, study different treatments. We use them in the food industry all the time to enhance the items that we ingest, or just to add additional flavor to them, or to produce bread. Something as simple as bread. You can't make bread without having yeast. So right there is a good example of how we're very highly dependent on microbes in our society. Bread, cheeses, olives, pickles, vinegar, yogurt, very common things that we use in pretty much all kinds of different foods that we eat. And then we talked about antibiotics already. So there's a high integration and use of microbial organisms in the biotechnology realm to make the world a little bit easier and a lot more convenient for us. In terms of different techniques that are out there in biotechnology, transferring genes using animal host organisms is kind of a common theme that you see. So this is kind of what you see in a lot of, sci-fi films. So we want to take a certain bacterial gene or a gene from a virus and then plug that in, or DNA from a virus and plug that into an animal model, which could be something like a rat or a turtle, or, well, I think turtle because I think it's Ninja Turtles, or maybe a snake or frogs maybe, but just a different host animal organism. Typically you want to get a host that's very similar to humans, which is why there's been a lot of studies, done on monkeys, because they're very closely genetically related to us, and how they react to certain therapeutic gene transfers would be give us a lot more information than, say, using a rat model, because our structures are much more similar and our DNA is much more similar. Some other techniques is culturing. On the diagram that's listed is culturing of plants from single cells. So you kind of want to generate, I guess, different cultures where you can essentially take cells and just simply grow them for whatever reason, whether you want to express a protein inside of them, or whether or not you're just interested and curious to understand further the cell structure. So different plants have, we've been able to culture a lot of different plants simply from single cells, because plants typically undergo the process of mitosis to enable them to keep growing. We undergo that too, but plants are also phototrophic or autotrophic, which means they can produce their own food. So you see a lot more kind of stability in terms of their capabilities, and they're not as complex as some of us, as animals, because of their cell systems. Other different areas, you've got essentially micromolecular biology and monoclonal antibodies that all kind of feed together. With monoclonal antibodies, these are produced where you can extract an antibody from something like a horse, and then you can replicate that in something like a B cell, which would produce antibodies in your body. And so once you extract that particular antibody, you produce it you could produce it and inject it into a cancer cell. And so that cancer cell will start to produce all of these antibodies, and it will massively proliferate and give you large quantities, because that's what cancer cells do. They massively proliferate. So if we can inject genetic material inside of those specific cells, we can have mass quantities of antibodies, and they would all be monoclonal, because that's how they produce naturally. So we can kind of tap into that and exploit a little bit, and in doing so, that'll help us to generate different diagnostic techniques as well as treatment techniques, and hopefully one day be able to treat cancer through the study of monoclonal antibodies. Molecular biology encompasses pretty much everything. In the biotechnology realm, we've got DNA technology, genetic engineering, the production of tracers, which are essentially just probes where you're probing for different portions in terms of segments of DNA, or in terms of how DNA is being identified. And so when we talk about tracers, we're talking about identification of ways to, kind of map out the genome and understand different locations of it. It also helps us to enable to figure out where different disorders arise in the genetic code. Genetic engineering, you have either production of different cell components, you can have production of different proteins, or you can essentially have, just cloning of cells, which will lead to large quantification of human proteins. You can also use it as a way to develop different human chemicals that might be needed in different systems, or in the production of different gene therapies. So genetic engineering is probably what you're most familiar with in terms of the underneath the biotechnology umbrella, because it's what we kind of hear about in the news all the time, is genetically engineering this, genetically engineering that. But there is a variety of other things, and also DNA technology, which can be used to help solve crimes. All right,